Hey, welcome back. So we had just finished part two of the lecture on chapter 15, innate immunity. And we talked about specific cells of the second line of defense, those 5 different kinds of white blood cells. 4 of them play a role here in the second line of defense. Now we're going to move on to this protein system that we have in the blood. It is known as the complement system because it's complementary. That is, it completes or provides more support to those cells that we talked about. What they have in common is that it's non-specific chemical defense against pathogens. That is, these proteins can act against lots and lots of different kinds of invaders. So like many of the proteins that are in circulation, these proteins are made by the liver and then released into circulation. And they're inactive most of the time. They're just present in case they need to be activated. And so it's this complex series of reactions that happen. So the first complement protein gets activated, and then it triggers the next complement protein to get activated, and so on and so on. And the videos I'm going to show you and that are involved in the homework, they're very intricate, and it's very easy to get lost in the details of, what number, what letter was that. And I'm going to say, let's not worry too much about it. The proteins all have names that start with a C for complement. And when they get activated, they turn into, there's a C for complement, there's a number associated with it, and there might be a letter associated with it. Just focus on the fact that there are these C proteins, and that we are going to see how they work together to do this big grand finale at the end of the complement cascade. But also individually, some of them have different jobs. Like some of them can act as opsonins. Remember, opsonin, all open mouth. It makes these microbes more attractive to the phagocytes. So it induces phagocytosis. Let's see. So we're not going to worry too much about the numbers on them. The grand finale is these complement proteins work together to they start like digging holes in the side of these bacteria and create these giant openings that allow water to rush in and lyse the cell. It's incredibly violent and so satisfying. And they can also augment the inflammatory reactions that are already happening. There are 3 different ways that have been discovered so far that can get this system going. And the fact that there are these 3 different ways indicates that there's lots of ways of kind of notifying the system that there's trouble on board. And some of the videos will show the different pathways. All right, so here's a diagram that illustrates the point. The activation of the proteins, the C for complement, and then an individual letter for a particular kind of protein, and then it can get activated, and then it can become, there's this other part to it. Don't worry too much about all the details. What I want you to see is the first way that it was discovered that this system got activated had everything to do with what we're going to talk about in the next chapter, the adaptive immune system. So this very specific part of the immune system that triggers very focused attacks produces something called antibodies. And antibodies themselves can sometimes induce this complement cascade to happen. Separately, there was another pathway that was found, and then a third one has been found, and possibly during your career as nurses, you may find that there's a fourth pathway that's been found. But right now we're at three different pathways. Um, but they all have in common that they activate the same set of proteins, and at every step that they get activated, it amplifies the signal. You get more and more and more of a reaction. Okay. So here's the video. It's going to cover these three pathways. Please don't get all caught up in all of the detail, because there's time for that later. What I want you to see is what they have in common.
Complement can be activated through three different pathways: the classical pathway, the alternative pathway, and the lectin pathway. The classical pathway was the first activation pathway of the complement system to be discovered. The classical pathway begins when a pair of antibodies attach to antigens, such as proteins, or a large polysaccharide on the surface of a bacterium.
Okay, I promise that I'm going to define these terms, but antibodies are things that we make in our bodies, and they stick to things called antigens. Antigen stands for antibody generator. It's a thing that can cause the antibodies to be formed, and the antibodies will react to them. Once you get a couple of antibodies stuck here, that can be enough to start triggering a series of reactions with these complement proteins. And so you're going to see these animations of things kind of flying in from the side and then breaking in two and then recombining. Don't get too upset about all those changes. What I want you to see is a bunch of reactions are happening so that other things can happen to fight off infection.
The antibodies bind and activate C1. C1 then cleaves several copies of C2 and C4. Together, the C2A and C4B fragments form an enzyme that cleaves C3, activating the complement system.
Okay, so what that all meant was some proteins activated each other, and now we're really going to get to the heart of what this complement system is about. More and more protein. What's not being shown is that at every step, there's a tenfold increase in the number of molecules that we're talking about. So yeah, so you start with a small reaction, and then suddenly you have lots and lots and lots of these little minion molecules out in the blood that are going to have an effect against the microbes. In other words, lots of different.
The alternative pathway of complement activation was so named because it was discovered after the classical pathway. While the classical pathway requires antibodies specific to the invading microorganism, the alternative pathway requires three complement proteins called factor B, factor D, and factor P, or properdin. These proteins are attracted to microbial cell surface material, such as the lipid carbohydrate complexes of certain bacteria and fungi. Together with C3.
Whoops, what happened? Come on. Where was it? Okay. Sorry, I will find our place. Oh, for crying out loud. Where were we?
Coating the invading cell.
I cannot seem to find our place.
Inflammation.
We'll just start there.
C3b acts as an opsonin, coating the invading cell. This coat makes it easier for the phagocyte to adhere to the invading cell during phagocytosis. C5b binds to C6 and C7, which attach to the plasma membrane of the invading cell. C8 and several C9 molecules bind to the C5b,
Boom,
C6,
boom,
C7
boom. I'm
complex,
gonna make a big
forming a
hole.
cylinder-shaped membrane attack complex, or MAC. The MACs form circular holes in the membrane of the invading pathogen, causing water to enter the cell.
Yeah, he's not excited enough. This is, I don't know if you saw this, but like this is this beautifully choreographed thing that happens with all these different proteins that come together. And so that's what I keep referring to as the grand finale of this whole process. Yeah, you get opsonization and you get these other things happening, but this membrane attack complex is hardcore. That is, you're going to dig a hole and then you're going to put all these posts around in a circle and bam, there's this big opening, and that's going to allow water to rush in and kill this cell. Just fantastic. Let's see, we're going to pause for a sec.
And ions to leave. This results in cytolysis, killing the cell.
Yeah, he's not nearly excited enough. The cytolysis, cyto, cell, and lysis, cutting or splitting. So busting the thing open. All right. Oh, and then here are a membrane attack complexes. This is a scanning electron micrograph showing these giant craters that have been made in the side of the cell. That should be terrifying if you're a bacterium. All right, and that is the end of part 3. We will continue over in part 4. See you over there.